Hello, everyone. Welcome to Foresight's Biotech Group. Today, we have Travis Potter with us. We'll get to hear about the work he's doing at Ovale on gametogenesis, which is the process of creating eggs from ordinary cells. So without further ado, this is babies from a blood draw. So exciting. Thank you, Travis.
Hi, everybody. Travis. I'm a co-founder of Ovale. Share my slides. Yeah, so I'm going to be talking about babies from a blood draw, the emerging science of in vitro gametogenesis, which is, as Lydia said, creating babies from ordinary cells. So the mission of Ovale is to empower all humans to have their own children, and we're doing this by making IVG a reality, which is the process to create eggs or sperm from regular cells, and then those egg or sperm can be used in regular IVF. So the big unlock of IVG is the process to create viable eggs or sperm from regular cells outside of the body in vitro in a dish. So take a normal cell, reprogram it to a stem cell, and then do this process of IVG, which differentiates those cells for normal cells to become human eggs or sperm. And IVG is going to be the biggest leap in reproductive technology since the invention of IVF in 1978, and will offer anyone the ability to have children regardless of age or fertility status. So a little background. So as all of you are aware, there's a fertility crisis in the world, and it's become one of the biggest challenges that Western societies and even sort of global societies are facing, where fertility has fallen below the replacement rate in almost every country in the world. Whoops. And government interventions haven't really worked very well, i.e. the baby bonus in South Korea. So we believe that probably one of the best way forward to combat this is to advance the science, is to allow more people to have kids when they want to. And there's a lot of big implications of this fertility decline: fewer workers, less innovation, more fiscal issues, and just more sort of less dynamic societies as the societies around the world age.
It's not because of infertility, though, is it?
It's not because of infertility, but it is because this is part of my story too, is that modern life has come sort of fundamentally misaligned with the fertility window of people. So 1 in 6 couples now struggle to conceive because, women and men want to sort of establish themselves in their careers, and they want to get maybe a PhD or whatever it is before they start having kids. But women's fertility still peaks in their 20s and declines quite rapidly thereafter, especially after 35. And this has led to a number of different consequences. For the first time in 2023, there were more births to women over 40 than there are to teenage girls. I mean, this is quite a change even in my lifetime. I remember in high school how they were really emphasizing that, the teenage pregnancies were a problem and how we need to get it down and whatever. And now it's totally reversed. Now the problem is that women in their 40s can't get pregnant or have a hard time getting pregnant. And this has also driven demand for egg freezing, which is up 40% 2022 and 2023, but has unclear efficiency. And a lot of these frozen eggs, frankly, are just never used. So about 1 in 6 couples now struggle to conceive. That's about 6.7 million women in the US are affected by infertility, but not all of that demand is even met because the insurance coverage for IVF is pretty non-uniform in the US. Some states mandate insurance cover it, others don't. And places where the insurance coverage is more uniform, like Europe, about 5% of kids are now born via IVF. And even outside of that, we estimate there are 800,000 women in the world who can afford IVF, but for whom it does not work. These can be like survivors of different cancers that damage their ovaries, women in their late 30s or early 40s who IVF just doesn't get enough eggs, or women with a number of other genetic conditions or other diseases. And this is the reason that I actually started Ovale. I'm part of this demographic story. My wife and I waited until our late 30s to have our kids, and we struggled with our second kid. And we went to fertility clinics, and, and I was like, man, this is kind of awful. Like the drugs and the semi-surgical egg retrieval, and then you only get so many eggs. And it was just like, why isn't there a better solution than IVF, which was invented in 1978? So it's quite a long time ago now, and we need to pursue better solutions. So yeah, current solutions like IVF and egg freezing are invasive, expensive, and not always effective. So we're seeking to try to solve these issues. So babies from a blood draw sounds quite science fiction, but will probably follow the path of many previous technologies. In 1978, after IVF became a reality, there was this term test tube babies, which was to refer to like kids created via IVF as like these fake humans that they know they're not real. But now there's been more than 10 million IVF births, and I think IVF is widely loved as a technology. When Roe v. Wade was overturned and IVF became illegal in Alabama, the legislature there voted unanimously to reinstate it. And so we think that there will probably be a similar path here where IVG will first start with women who have no other option to genetic parenthood except this, but will eventually encompass all of IVF because it will end up actually in the long run being cheaper. And this is because IVG does not involve a semi-surgical procedure. It just involves a blood draw and then a sort of cell culture process. Whereas IVF also involves this semi-surgical procedure. So it's actually harder to get the cost down on IVF than IVG. And another data point here is, when ultrasounds first came in the 1990s, people thought they were necessary. Now it's sort of this sort of responsible parenting threshold, and 97% of people get them. So we think that the same thing will happen over time with IVG. So let's get into the nitty-gritty process of how does IVG work. So we're taking somatic cells or regular cells and we're reprogramming them into stem cells. This is by the Yamanaka factors. A Nobel Prize has been awarded for this. Well understood. Then we take those stem cells and we differentiate them into two types of cells. One is on the top here are ovarian supporting cells, or the other types of cells that are found in the ovary. And the reason we need ovarian supporting cells is that these eggs that we create on the other side start out microscopic and they need to get to macroscopic. So we need to grow them 100x in size. A mature egg is the only the only cell in the human body that you can see with your naked eye. So we need to create sort of this nurturing environment, these sort of synthetic ovaries capable of producing eggs. So we create ovarian supporting cells. And then we also have to create the eggs themselves. And there's two big things that we need to do to create them. First, we need to do epigenetic reprogramming. And so these somatic cells and stem cells have quite different epigenetics than germ cell. And in general, one of the big things in germ cell specification is the extensive epigenetic reprogramming that they undergo. So we need to recreate that in a lab. Then, once we have the proper epigenetics, we need to induce meiosis, or the special type of cell division, where the resulting cells have half the chromosomes they usually do, as you get half from your mom and half from your dad. And then we need to reconstitute these synthetic ovaries, we call them ovaroids, that are capable of producing eggs, and then mature these to M2 oocytes that can be used in IVF. As some of you are aware, this immature egg to M2 oocyte, this is another company, Gametogenesis, and currently in phase 3 clinical trials in the US demonstrating this technology. And they are actually using technology that was co-developed by our CSO, Merrick, during his time at the Church Lab at Harvard. So he created adult ovarian supporting cells. And so they culture these immature eggs to mature M2 oocytes with those cells because this happens in an adult woman. However, the reconstituted follicles, or the follicles, are created in the fetus. As you know, a woman is born with all the eggs she'll ever have. So the first part of this, we need fetal ovarian supporting cells, which is different than the ones that Merrick developed during his PhD.
Excuse me, may I ask a question?
Mm-hmm.
So if you take any old cell out of my body, well, won't they have exactly the same genetics, unlike egg cells and sperm cells?
I'm not sure what you mean by exactly the same genetics, but we can. Sorry, go ahead.
Every white blood cell in my body has exactly the same genetics, unlike my sperms.
Right.
Whatever are left.
Yeah. So we need to trigger the process where your cell undergoes meiosis, and then it will have, it will be like a sperm cell that, just like your body makes sperm cells, right? It can trigger the same sort of program, and then from a normal cell create sperm.
I know, but if you had 100 normal cells, you would arrive at absolutely, well, I guess you'd have exactly 2 types of cells that come out, not hundreds or millions or whatever. I don't know what the number is. But in other words, every egg has different genetics. unlike these cells you're making. Is that
Yeah,
correct?
yeah, because
Okay.
that's correct. These should be indistinguishable from sort of natural sperm. So like, yeah, like you said, like every sperm has slightly different genetics because of the crossover and recombination that happens during meiosis.
Okay.
Great. Let's save the questions for the end of the presentation. Thanks.
So healthy mice were first demonstrated from this IVG process in 2016, but probably the most relevant paper for us is this generation of ovarian follicles from mouse pluripotent stem cells and Yoshino et al. In 2021. And this here is a picture of the healthy mice that were created from IVG. So they took mouse embryonic stem cells and they did the procedure that I described before, where they differentiated some of them to ovarian supporting cells, they differentiated some of them to germ cells, they reconstituted them together, and they got healthy mouse pups. But the one thing to note here is that the efficiency of this protocol was between 2 and 5%. That means between 2 and 5% of pregnancies became live birth, which is lower than what you would get in, say, IVF, which is more like 30% or 20 to 30%. And the reason is because the epigenetics on these resulting eggs were not always completely correct. However, the mice that were born actually seem ironically healthier than normal. And this is because mice usually have babies in litters. And since the efficiency was low, it was usually singleton births. And so the mice, got more sort of nutrients love from mom than a normal mouse pup. So there are three sort of approaches currently being worked on for IVG. The one that was used in the paper before and the one that was demonstrated first is this sort of signaling and cytokine approach. And this one sort of tries to mimic the body's natural signaling environment in a dish. Everyone believes this will work because this is the process that happens in women, and almost all women produce eggs. And also it was the process that's been demonstrated in mice. But it has a number of drawbacks. One is, I guess, the obvious fact that it's proved exceedingly difficult to translate to humans. This was first demonstrated in mice 10 years ago, and nobody has yet created a egg cell for a human from a human cell line. The second, and part of the reason that it's taken so long, is that there's just a long timeline from the original somatic cell or stem cell to an egg. If you exactly recapitulated the signaling environment in a woman's ovary, then this would take something like 14 years, because that's when a woman first starts ovulating again and maturing egg cells. Obviously, you can fast forward a little bit through that and skip sort of the quiescent stage in the middle. But even if you do, it might take as long as 12 months to go from a regular cell to an egg, because there's so many different steps that you have to go through, and all those processes are not particularly fast in the body. The other thing is that these cytokine or signaling approaches have shown they're hard to replicate. For example, the cytolag protocol in the mice, the one that we showed before, has only been replicated by a few labs around the world, though now there are a few more. And that's just because they're very tricky and there's a lot of variability in getting the timing exactly right and things of this nature. However, there are a number of people working on this approach, including, of course, the Cyto and Hayashi labs, which first demonstrated this in mice. They're trying to translate it to cynos in humans, as well as another IVG company in California called Conception. The other approach that you might have seen on social media recently is what I would call the cloning approach, which is basically take a nucleus from an existing cell and implant that into an egg and then sort of hope meiosis works. I guess the advantages of this are that it's relatively simple. It relies on cloning techniques that have been around since the 1990s. There's a number of quite strong problems with it, though. One is that it's a low scalability. It's because it requires donor human eggs. Not just one egg, it requires many eggs, because this sort of cloning somatic cell nutrient transfer has quite low efficiency. So you need something like dozens or hundreds of eggs for every embryo. The second thing is that it scrambles the DNA. So it seems like eggs are, want to do meiosis too, but most of the important part of meiosis is meiosis 1, and they sort of skip over that. So the DNA gets scrambled. So we think that this is quite unlikely to ever come to market as a therapeutic, but there was a research paper by Palamato and Stipanov and others that went somewhat viral on social media at OHSU. Finally, we're working on the third approach, which is a transcription factor-based approach. So we're trying to differentiate stem cells towards specific cell types by introducing transcription factors. This is a number of advantages. The first is that it's much faster. Some of our protocols take 1/10 the time as the signaling-based ones. This is because we can fast forward through or skip some of the intermediate cell states. And the other thing is it tends to be very highly replicable. Sometimes the transcription factor protocols not only work across cell lines, but even sometimes across closely related species. So this will allow us to bring therapy to a much more diverse patient population much more quickly. There are some disadvantages to this approach. Historically, transcription factor overexpression has been known to create hybrid states where you sort of get epigenetics of the parent and sort of daughter cell type, and you can get these hybrid states. However, I think in IVG, we get somewhat lucky because we have to do this epigenetic resetting. We erase most of these epigenetic tags, and so it's much trickier to get in a hybrid state since we've erased most of the state to begin with. This approach is also relatively new, and not many people have worked on it. Merrick, who's our CSO and co-founder at Ovell, is one of the few in the world who's tried this approach. But we think that the likelihood that this works in the next few years is quite high based on our progress so far. So to go a little bit more into the two approaches of IVG, the signaling method is on the left here, sort of like this mouse, goes through these certain steps to create eggs, and then you're just going to try to replicate exactly these steps in the signaling environment in a dish. So go through all the different steps. Whereas the transcription factor-based approach is much more like trying to find the cheat code, trying to find the gene regulatory network that drives the cell to become the target cell type that we want. This has the advantage of sort of being much faster than the other approach. The other advantage of the transcription factor-based approach is that they're much more amenable to computational screening. So we can take these gene atlases, we can infer regulatory networks from them, gene regulatory networks, and then we can rank the factors that are most likely to be driving cells towards the specific cell fate that we want. We can clone those in the plasmids. We can then dump those plasmids into a pooled screen in stem cells. So say we got like 100 transcription factors, RNA binding proteins, or other things that we think might drive cell fate. And each of these has an associated barcode. So then the cells differentiate. We can sequence all the cells. We can determine what transcription factors integrated with what cells. And then we can also match these cells back to the gene expression data to see what transcription factors are associated with the cells that are close to the cell type that we're looking for. And then we can sort of iterate. So this is like a much more engineering-based approach than the cytokine type approach, which is much more guess and check. Like, I think this environment's going to work. Let's try it. I think these chemicals are what's present. Let's try it. So this allows us to do much more high throughput, high scale type screening. So Merrick's, our CSO's previous work at the Church Lab, he used this technique to create a number of different cells in IVG. First, as we talked about before, with the adult like ovarian granulosa cells that have since been licensed to Gamito. And this picture on the left is them self-assembling into a follicle-like structure. So these are all the supporting cells, and then the middle is where you would expect a supported egg to be. An egg's not in this picture, but it did make the hole for it. The other thing he did was he was able to induce meiosis in stem cells. So this is a picture of the middle of a meiotic cell. And then on the right is showing that this meiotic cell demethylated quite quickly, but did not get to the sort of 8% global CpG methylation that would be associated with germ cells. So a promising foundation, but lots of work to do. So we have solved a number of these problems since we founded the company, including solving the demethylation problem before. We have a world-leading rapid and complete demethylation. We've enhanced the meiosis induction protocol. We're the first to show in vitro intercellular bridges. So it's known that intercellular bridges, both in mice and humans, precede meiosis. So we've been able to sort of demonstrate that functional readout. And we've been able to create these fetal stage ovarian supporting cells that match the stage that we need to support the first stages of meiosis. So epigenetic erasure. So before in the field, in the Cyto lab, demonstrated slow and incomplete, or we demonstrated incomplete methylation erasure, and Cyto demonstrated slow methylation erasure. Their protocol that's published takes about 120 days. In the body, there's not a lot of data, but because these cells are both rare and sort of inaccessible, but it takes about 60 days in the body. And our protocol that we have developed takes about 28 days. So it's about 2 times faster than the in vivo and about 4 times faster than the in vitro state of the art. And I guess importantly also, this demethylation, when it's compared to the in vivo data sets, as close to the in vivo data sets as the in vivo data sets are to each other. Then we optimized our meiosis protocol. So the protocol at the Church Lab had about 20% efficiency. It's in microscopy where we have markers on different meiotic genes. And then on the right, you can see that it's much more efficient. So we've gotten up to about 70% efficiency now with a simplified and improved protocol. Finally, we've demonstrated intercellular bridges. So intercellular bridges are known to precede and support meiosis in mice in humans. And this is in human cells in vitro. This is TEX14 down here, which is a very specific marker of intercellular bridges. And you can see the little ring between the cells. Intercellular bridges form when cells do incomplete cytokinesis, or they don't completely divide. So they form these germ cell nests where there's multiple cells that are connected by these incomplete divisions, and they sort of pump cytoplasm and other nutrients to the cell that's going to become the egg. So they're like the supporting cell. And our intracellular bridges also match sort of the statistical properties of in vitro intracellular bridges. So we get like roughly the same number of germ cell nests that have 6 cells or 8 cells, what have you. And on the left here is a zoomed out version of this TEX14. These little puncta here, these little bright dots are all little intercellular bridges that the microscopy is picking up. Next, as I mentioned, we created stage-matched ovarian supporting cells. So previous attempts, like Merrick's attempts at the Church Lab, created adult ovarian supporting cells, and these were not appropriate to support meiosis, which starts in the fetal ovary. Women are born with all the eggs they will ever need, and at a diplotin arrest, which is partially through meiosis. So we need to create the appropriate supporting cells. And they also, you can see here, they reconstitute into sort of the same follicular-like structures as the adult granulosa cells. One of the things that we talked about before is this idea of somatic cell mutational load. So in IVG, we have to be careful about which cells we're using as inputs. That's why the title of this talk is Babies from a Blood Draw, as opposed to, say, babies from skin cells. And the reason you wouldn't want to use skin cells is because they have quite high mutational load, much higher than germ cells in the body. And in particular, you wouldn't want your baby to have some weird mutation because you had a sunburn 10 years ago. So we've calculated that with the proper cell types and the proper controls, that we can actually get the mutational load down below that of natural sperm or eggs. All sperm or eggs have some mutations, usually on the order of about 100, and we actually think that with careful selection and sequencing that we can do better than that. The other big concern with IVG, as we talked a little bit about, is making sure the epigenetics are correct, and that's part of the reason the efficiency in mice right now is low. But we think that over time, we can actually ensure that the epigenetics are better in our cells than natural cells. Only about 1/3 of eggs can even create a viable embryo because of different defects, often with the epigenetics with the eggs or sometimes with the genetics. And we can check all these things prior to this to make sure that we are only creating the highest quality, safest cells. So what makes a good egg? What goals does IVG need to hit? First is chromosomal. So they need to be arrested in metaphase 2 of meiosis. They need to have the proper number of chromosomes. It needs to go properly through crossover and recombination. Second, as we talked a lot about, it needs to have the proper epigenetics, including the proper female imprinting. There are some known imprinting disorders like Angelman syndrome, and we want to make sure that we have a good enough protocol that's not a risk. And finally, cytoplasmic. The eggs need to be of sufficient size and have the correct proteins and RNAs that they can support embryo growth. Finally, a little bit about the regulation. So different countries are setting up different regulatory paths for IVG. There's been discussions in Japan and the UK and the US about how to regulate and how to bring IVG to market. So the FDA had a meeting, the National Academy of Sciences, on IVG. And in it, they said a number of different things. First, they claimed authority to regulate IVG as a biologic cell therapy. Second, they said they wanted to see data that proves that the genetic and epigenetic integrity of IVG-derived gametes and embryos are comparable to their in vivo counterparts, which I think is a quite reasonable thing. And then Third, they want to see multi-generational studies in rodents to make sure that IVG doesn't, excuse me, doesn't do anything strange down the generations. And they also want to see demonstration of healthy offspring in non-human primates. And the other thing that was discussed briefly was this rider in the FDA. The FDA cannot take a trial that has the intentional heritable genetic modification of any germline. However, IVG doesn't fall under this rider because there is no genetic modification. We are merely by triggering the processes that already happen in the cell. We're not inserting or doing anything with the gene. So it's just epigenetics and not genetics. The first birth from IVG is likely to be outside the US. This is probably an analogy with gamete. Their first baby was born in a fertility clinic in Lima, Peru, and it's since been legal for a while in Mexico, Peru, Australia, and I think some other countries. And IVG will probably be the same way, where US trials are more extensive and some other jurisdictions are willing to bring it to market slightly faster than the FDA. However, we believe that if IVG is proven safe and effective, the FDA will approve it, especially as it's the only option for some parents to have biological children, and it really is a treatment for the disease of infertility. So our roadmap right now is to refine the processes that we currently have and demonstrate a human egg in the next few years. And then we want to take a few years to demonstrate the safety of that egg, including optimizing the protocol to make sure that the efficiency is high and that the epigenetics are correct, and create a quality control platform around that, as well as do the enabling non-human primate studies that the FDA and frankly we would like to see before we have the first human baby. And then after that, it will move it into trials around the world, and different regulators will move at a pace that is comfortable for those countries. And like I said, as we expect that there will be different speeds in different places of the world where people approve IVG. We can see in particular that places like Japan or South Korea may want or may end up being more aggressive bringing IVG to market because their fertility crises are even much more dire than the US. I think in South Korea, it's something like 10% of births are now IVF. So they have a huge issue there. Yeah, so that's my presentation. We're looking for a future filled with joy, and every family deserves the chance to experience the unconditional love of a child, and that's what we are trying to do.
Thank you so much. That was a great presentation. I think we have a couple questions. Some in the chat were just debating the ethics of overpopulation. However, maybe we can move to the first question from Christine. Christine, would you like to ask it? Or if you're not available to speak on a mic, I can also ask it for you. Yes. Okay, I will ask it. How far away are you from examining human embryos created by IVG by PGT testing? Given that the issues are epigenetic, would PGT testing even correlate with miscarriage rate?
So human embryo is probably a little bit away. We're going to demonstrate an egg, and then we'll move into non-human primate studies and have some non-human primate babies. We want to create as few sort of embryos, human embryos, as possible that aren't actually going to become children. Yeah, so I think that PGT and PGA and some of these are tests that we'll definitely do, but not for sort of the more dramatic things that you see on social media, but we just need to make sure that the meiosis was done correctly and that there's no aneuploidy and things of this nature. In addition, we will also need to do sort of epigenetic tests, like a bisulfate test, to make sure that the epigenetics are correct. So I would say that these egg cells and these embryos are going to be some of the most tested cells in sort of history when they go into humans. So any sort of test that you can imagine will probably be run.
Awesome. We have a couple more in the chat, but maybe we move to Micah and then Robert.
Just a quick background question. Does IVF with a young egg and an older woman work well, or do we also have to solve the problem of an aged womb along with the aged egg?
Right. So IVF generally works much better if you get a young egg and you implant it in an older woman. In fact, I think the oldest birth that some woman had with a donor egg had a kid in her 70s, which, clearly not possible with her own eggs. But the reason that the eggs decline in quality is generally that the proteins that in the eggs decay slowly over time. So there's a lot more decay in these proteins that are binding together the RNA than in younger eggs. And so when we create these eggs from IVG, we won't have that problem, because when you create stem cells, even stem cells from an older person, and they undergo this sort of epigenetic resetting, they look exactly like cells from a younger person. So it shouldn't really be an issue from that perspective.
Great. Awesome. Robert, would you like to go?
Yeah, sure. Fascinating presentation. Thank you for speaking to us. This is great. I had a couple of questions. The first one relates to mitochondriogenesis, and the second one relates to the applications of this for the production of, essentially as a route for the production of replacement organs. So the first one, I guess, would be, my understanding is that oocytes contain vast quantities of mitochondria. And I had looked a little bit at in vitro gametogenesis as a potential route to the production of large numbers of mitochondria. My first question is just, am I correct in thinking that that these cells produce vast quantities of mitochondria? And if so, have you thought about, do yours also generate large volumes of mitochondria, and would it be a good source for that?
Would it be a good source for mitochondria? To be honest, we haven't really thought about it as a source for mitochondria. You are correct that, an egg cell would have like vast numbers of mitochondria. They go through this like cleansing process in creating the eggs, though, where they try to purge all the bad mitochondria, because you also build up like mitochondrial mutations, and some of the mitochondria, just the proteins break down. And so they try to purge all those bad mitochondria. So it goes through like a very narrow bridge, but then there's like an explosion, in the number of mitochondria. There's like thousands and thousands afterward.
So in your gametogenesis, does that same quality control of mitochondria take place?
Yes. Much of it takes place actually in the reprogramming of somatic cells to iPSC cells. It kind of mimics this bridge. But then we will also have to do sequencing on these stem cell lines and quality control to make sure that we are selecting a stem cell line that has healthy mitochondria. There's actually been a number of papers that show that since the distribution of mitochondria in clonal stem cell lines is non-uniform, that you can actually find, daughter cell populations with very good mitochondria. In fact, this will actually be able to cure severe mitochondrial disease in a way that's legal in the US, because right now the FDA has said that this 3-parent baby, the mitochondrial replacement therapy, is not legal. But when you have all these clonal stem lines, then it's just selection. There's no genetic modification. So it should actually be able to cure mitochondrial disease.
Okay, excellent. And then that second branch of question was whether or not this might be a way for an individual whether the work you're doing might lead to an alternative path to the production of replacement organs down the line.
Sorry, go ahead.
Basically, the thinking is, if I'm female, I have eggs, and I could potentially, use in vitro gametogenesis to come up with sperm, and you're building a platform that it seems like might also be suitable for the production of a twin of me that has organs, and I might be able to tinker with it to provide an ethical source of organs, as long as certain precautions are taken and genetic changes are made.
Yeah, I don't think our technology will be used to do that, just because I think there is a lot of sort of ethical guidelines around, actual embryos, which is what we would be able to create. But I do know there is some other companies like Renewal Bio who's trying to take stem cells and then sort of activate programs to make sort of like organoid type systems from.
Great. I think there were 2 others that had raised hands. Aaron and then Micah.
Oh, sorry. Yeah. No, I came into the presentation late, so I didn't see the whole thing. So I was wondering if he had spoken to Omri, and he just mentioned Renewal. And so I'm sure that he's aware of what they're doing over there. So that's all.
Oh, yeah. Omri would be a great person to connect with. Good point. Okay. Micah, hit it.
Can we already generate sperm using the same technique? And is it much easier, I'm guessing?
Yeah, so So actually, there is a company, Paterna Biosciences, which is trying to take spermatogonial stem cells and then, so it's like a sort of intermediate cell state, and then create sperm from them. And it does look like this is going to be easier in men than women. And I think there's sort of two reasons for that. First is that men have, an X and Y chromosome. So you don't really need to do any epigenetic reprogramming beyond the normal epigenetic reprogramming that happens for that. Whereas women have two X chromosomes. And so in the stem cells, one of them is inactivated. And so you need to reactivate the X chromosome as part of the epigenetic reprogramming. So it's slightly more complicated. The second thing is that sperm cells don't really need to be grown to large size. And so you don't need to keep them in sort of tightly controlled culture for as long. Whereas, here we need to grow these cells up 100x in size. And so, yeah, it does seem quite likely that IVG in men will end up proving easier than women.
Awesome. Does anyone else have any other questions? There are some in the chat as well. Well, John Hemming asks, what makes a good egg, with the context that mitochondria are not mentioned? Is that because it is thought that they don't matter? Maybe we touched on that already.
Yeah, so we touched a little bit on the three criteria that are sort of necessary and sufficient for a good egg, which are, the proper chromosomes and genetics, the proper epigenetics, and the proper cytoplasm. I guess mitochondria were kind of rolled up in the proper cytoplasm. But yes, I agree that mitochondria are important, and it's been shown to be increasingly important in health and conditions for all this sort of stuff. And it's something that we would put as part of the quality control platform, both at the beginning and selecting the stem cell lines, and then at the end to make sure that the eggs were healthy and correct.
I love the conversation that's occurring in the chat on overpopulation, that we don't bring it into our Q&A, but it's there. Micah, would you like to go?
Yeah, along the path to what you're trying to achieve, do you think you'll pass over reliable mouse generation using this technique? It would be nice to be able to do transgenic mice using this technique instead of the current techniques, but it would require getting to, much higher reliability. Do you think that's on the path, or is that kind of a separate branch that you're not taking?
It's not on our roadmap outside of satisfying sort of the FDA desire to show that IVG is safe over multiple generations in rodents. But maybe that work can eventually be used by others to do sort of transgenic mice and have a much higher efficiency system in mice. But it's not currently like on our roadmap to improve the mouse efficiency.
I second this interest, and not necessarily from an efficiency standpoint, but from a timeline. If you're able to produce a novel transgenic mouse line in, a couple of months instead of it taking a year to do all the intercrosses and everything that you need, that could be very helpful for research.
If I may ask a question. Oh, sorry. If I may ask a question, can you make eggs from male cells?
So this has been demonstrated in mice, actually, that there was a birth from two male mice where one of them went through IVG and they were able to sort of get rid of the Y chromosome and create an egg and then fertilize that egg and put it in a surrogate and have healthy mice. And then there was another group in China that was take sperm from two different mice, edit the epigenetics of one of the sperm in an allele-specific manner, and then fuse those two sperm into an egg and have a mouse that way. So it is in theory possible. Right now, it's not on our current roadmap, and just because the need is for eggs initially.
Great. Has a question in the chat. I can ask it for you, or you can ask it yourself. Well, maybe I'll just go for it. It's given that human germline cells have the lowest mutational burden of any cells in the human body by an order of magnitude or more, how do you ensure that the genome of your starting material remains high quality?
Yeah, so we touched on this a little bit before where, the first thing we do is we're going to be doing this from cells in the body that are known to have a lower mutational load. You wouldn't want to use skin cells, again, because they have a high mutational load, they're exposed to the elements, so you don't want any weird mutations. So that's step one. And then step two is to sort of do a lot of sequencing so we can identify what are the mutations and where they are and do that quality control. And actually, that sort of sequencing analysis is something that Merrick, our CSO, developed during his PhD in the Church Lab. So we already have it. And based on that, we actually think that, yes, these somatic cells have higher mutational loads on average than germ cells, but with all this quality control and careful selection of which somatic cells, we can get the mutational load to at or below natural reproduction.
Awesome.
To follow on that, is this, do you expect the sequencing costs are going to dominate the costs of this, or are there techniques to get those down? Because if you do like an off-the-shelf sequencing for $300 and you've got 10,000 eggs, that's a lot of money. But I'm guessing there's other techniques. Are there techniques that can get this cost down so the sequencing is not going to dominate it?
Oh, I actually think that you're correct, that basically the sequencing will be the sort of main cost in the future. I don't think we're going to have to sequence 10,000 eggs, hopefully, more, fewer number than that. But certainly that will be the biggest fixed cost when this comes to market.
I'm a little outside of my wheelhouse here, but doesn't the epigenetic age turn to zero during gastrulation? Is it like, I guess it's a couple of weeks after conception? That clears up, I guess that would clear up a lot of epigenetic detritus, if you will.
Yes, I think that's true. But like I guess shown in the mouse studies, like you need to get the epigenetics still close enough that sort of the program runs.
Okay.
Or otherwise it doesn't run at all and you don't get a baby. But I think I sort of second the point that like the mice that are born seem perfectly healthy. So,
I guess with the question on the cost of sequencing, Christine asked a question about speculating on pricing. I wonder if that has already been answered, or I guess she wondered, currently one round of IVF is $30K. Is this going to be a few fold higher or log scale more expensive?
Yeah, so I think it will be much more expensive when it first comes to market, probably like $200,000 or something like this. It partially depends on, like quality control constraints from regulatory bodies and things like this. But it will actually over time, over say, 5 or 7 years, fall below the price of IVF because IVG, unlike IVF, is a manufacturing process. So you sort of take cells and put them in one end of the machine, and the other end of the machine, you get eggs. And those things, fall to sort of the sequencing costs and the input costs and things like this, which is below IVF. IVF will probably always be a little bit expensive because it's a surgical procedure. So you got to go to a doctor's office. They got to, stick a needle in your ovaries and extract eggs. And that'll always be more expensive in the long run than manufacturing. But in the short run, it will certainly be more expensive. I think it's going to fall like most technologies, like, when they first come to market, they're quite expensive and then their price goes down over time.
Sure. Awesome. Okay, next question. Aaron.
Yeah, this is a bit of a weird one. Travis, do you have kids?
Yeah, I have 2 kids.
Okay. That's the question. That's all.
Yeah, that's how I got involved in this. I was trying to have my second kid, and it wasn't so easy. And I was going to
Oh
IVF
yeah,
clinics,
sorry.
and
You mentioned that.
Yeah.
Sorry. Yeah. Okay.
yeah.
All right, Micah.
I'm curious, since you obviously are very well researched in the subject, do you think that long-term this strategy is superior to just skipping to an embryo, like skipping the egg and just trying to generate an embryo out of iPSCs or something as a strategy? And if you them, like, contrast them, like, what's the difference? What are the advantages?
Yeah, I mean, if you're creating an embryo from an iPSC, it's much more like a clone, I think. So, you'd at least need to go through meiosis if you wanted to have sort of kids as opposed to clones. So,
just thinking about like for the generation of humans, and if you don't really care where the source material is, are they comparable? Is one easier than the other?
That's a good question. I think it's a little bit unclear. If your goal is just the generation of humans, Renewal Bio has been able to create these sort of embryo models, and they've gotten quite far, but nothing that looks like a human yet. And then IVG obviously also hasn't created a human, though it has created mice. So I think it's a little bit TBD. I'm not entirely sure.
I guess maybe I have a question, but it might be outside the scope of this like immediate topic area. But you mentioned earlier in your presentation about the progression of different reproductive technologies. And in your graphic, there was around, I think, 1978, you said IVF was invented and over 10 million IVF babies have been born, formerly considered test tube babies. I wonder, are there any like downsides or like risks that we see right now that are happening because of IVF as opposed to like conceiving traditionally?
Yeah, it's a good question. So there have been some studies that show that there's like modestly higher levels of like obesity or heart disease or things like this associated with IVF babies than with naturally conceived babies. But I think it's still a little bit unclear whether that's it's confounding or not. Like basically people that are doing IVF because their natural reproduction isn't working as well as most people's, and is that the reason that we're seeing like these modest effects or not? So there have been some slight effects detected over sort of the 40 years, but it's still really unclear whether you can even attribute it to IVF or not.
Okay, thank you. All right. Are there any other questions? We're nearing the end. Okay, cool. Josh.
I just want to put in a word for adoption. I adopted my 2 girls 36 and 40 years ago, and it was a joyous experience. I don't have the experience of a child with my own genes, but it's completely full, and it's the unconditional love that you described, having adoptive children.
And that's great. I'm glad you adopted it. It's wonderful that you were able to make a family that way. Congratulations.
Christine has another very juicy question. Do you have any idea why there is such a decrease in female fertility at 40? It seems that there is a gradual decrease and then a cliff in egg quality. What is that all of a sudden there? What is that all of a sudden there is such a steep drop off?
I can speculate a little. I want to preface it that I'm not actually an expert in this particular area, but it seems like the proteins that are binding the chromosomes together decay over time, and you get much higher rates of aneuploidy or sort of abnormal chromosomes than cells. So the thing with women's fertility is that you're born with all of the eggs you'll ever have, but then those eggs are sort of in this quiescent state for a very long time, and they start falling apart as they get too old.
Thanks.
Well, the environment also is degrading for, the incubation, if you will.
Building on that answer, has there been any work to try to rejuvenate the eggs directly without rebuilding?
Yeah, it's not my company, but there is another company. I can't remember the name right now, but they actually recently announced that they have a therapy that could potentially rejuvenate older eggs.
Awesome. Well, thank you everyone for joining. What a fantastic presentation. Thank you, Travis. Really
Thank you so much, Lydia.
To see your progress moving
Thank you so
forward.
much. Thank you everyone for coming.